Okay, welcome to week 6, sorry, of Advances in Omics. So, till now we've talked about sequencing technologies, different types of other, like other than genomics, we've talked about transcriptomics, proteomics, and so on. Today's session is basically mostly uh, navigating these data sets using Linux commands in general. So, just a second. I want to give you a. Just let me stop sharing once. Yeah, yeah. So today we'll talk about different Linux commands. So this is a drive link which contains this folder. Inside this, there are uh, some like test data sets. So you have VCF files here, you have BAM files, FASTQ files, and so on. So that you can, if you want to try this out in your own system, you can do that. So these are basically East genome files, right? So yeah. Uh, yeah, so we'll talk about different types of Linux commands and how to navigate. It's mostly a hands-on session today. Uh, I'll be showing some navigating through some directories in Linux and so on to let you know how it works majorly. But yeah, I do not have kind of like so. If you want to work on the data itself, I think you have to download some data sets from the internet and work on those. I do not have anything in my repository currently uh, to show you working on those, but I'll definitely try to show whatever I can. Right. So yeah, now let's get started. So let me show you a Linux terminal first. Right. So we'll open a new window. So mine is a MacBook, but if you have Windows, the way to access uh, Linux terminal is using the WSL, right? So you can take a look into this. In this one, if you install WSL, you can use Linux commands on WSL, right? So the normal Windows commands, Windows terminals won't support Linux commands. So if you want to do that, you can try using WSL, or there's something else called MOBA extern. So this is basically if you want to connect to some SSH client, right? So some external Linux system if you want to connect, this works well for that. So uh, if you want to do that, yeah, feel free. Okay, so this is a MacBook, so I do not need other softwares to kind of connect to a Linux terminal because the terminal on Mac basically supports Linux commands. So the basic command is ls. So this works, and every other kind of mostly Linux commands work here. So firstly, I would want to uh, connect to a system which has the data. So in general, this is how it happens. So you have another system where all the data is stored. That is probably a Linux system. You would want to connect your computer to that system and then uh, do work on that, right? So the basic way to do it is to use SSH clients. So I have a system in my department where I have an account in. So I generally connect my computer to that system, and then I kind of use my computer to access all the files in that system and kind of run codes on it, different pipelines, and so on and so forth, right? So, firstly, I would like to show you how it connects. So, the command is SSH, then your name, and then the IP address. So, in this case, this is my IP address here. but also you need to be connected to the same network as your Linux system so that you can connect to this. Mm, uh, did I do wrong somewhere? One second. Sorry. Yeah. So it asks for my password. Once I give my password, it gets connected. Now I'm logged into this particular system. This is a system in my department I have access to, but yeah, generally, uh, wherever you'll be working on in academic settings or industrial settings, anywhere, this is how it is done. So you have a system where you have all the data stored, all your scripts, all your software is installed. Because these are like big data sets you'll be working with, so you cannot run on your own computer. You have to connect to a Linux terminal and then go on about it. Okay, so this is how you connect. So now I am in a Linux terminal, right? So uh, let me start with some of the commands, right? So these are very basic commands. First is mkdir, pwd, cd, and ls. So these are the commands that we use on a very daily basis across, you know, irrespective of what you're doing. If you're handling a Linux system, these are commands that you'll be using very frequently. So make mkdir is basically, first let's go to my space. So changing of directory is done by cd. Cd is the command. um, then let's go to my place, downloads. Yeah. So this I have changed the directory. This was my home directory, and then from the home directory, I changed to this particular directory using cd, right? Then if I want to check out the files in this directory, the way to do is using ls. So here you see I have a couple of FASTQ files, some temporary other output files, so these are folders, and these are kind of other other files that I have. Now, if you want to sort them by time, and basically, if you do this ls-lt, it will uh, arrange it in on the basis of the kind of the latest edited folder or file will come on the top, and the oldest file will come at the bottom. If you do LTR, it kind of switches it. So the oldest file comes first, the newest edited file comes on the bottom. Right. So CD and LS are very important commands that we use on a daily basis. Now there are some caveats with CD. Though CD, after CD, if you use this curly hyphen. So this means the curly hyphen in Linux means home. So if you do that curly hyphen, it will take me back to my home. As you see, this was my home area. Previously, I was here. I did cd this, and I'm back to my home. So here also you can see this symbol. So this is basically the indicating of the home area. Now let's go back to there. Now let's check out what files there are. So these are like all the files that are present, and uh, I'm sorting it on the basis of time and last when it was edited. Excuse me. Now another important command is pwd, which is basically what directory you are currently in. In some cases, this part will be truncated, so you cannot see what directory you are in from your command line. So you need to do this command pwd, so it will tell you what uh, area of the system are in, right? So this is okay. Uh, Now let's talk about mkdir. Mkdir is basically make directory. Mkdir, say for example, I want to do test directory. So the command is mkdir space the name of the directory you want to create and enter. So now if you check the ls, you have another directory present here, which will be empty. If you do this and go inside, it's empty. So this works fine as well. So these are very basic commands, and cd also has this. You remember I told you this goes to home, right? Now let me go back to this. Right. this is the directory where there is nothing, right? Now what I want to do is I want to just go back to the parent directory, meaning I want to go here, right? At this particular location, not meaning just the parent directory of the directory I'm currently in. For that, we do cd space double dots. So it gets me back to this particular directory. So there are some caveats, some like kind of interesting things with cd. So this particular hyphen is basically home directory. Space dot dot is the parent directory, right? So that is there, and all the commands that you have. Say for example, sorry, uh, it tells you basically what possible options are there for your command. So for let's actually I'll tell this to you when we talk about a particular software. But yeah, this is how it starts. Now, moving on to some more uh, uh, commands. So for file creating and editing, so this was for basically just navigating the directories, finding what directory you are in, and so on, changing the directories and so on. Now we talk about file creation. So I'll talk about Vim, but Nano is also in a similar fashion. But let's talk about Vim. So Vim is an editor, right? So you type Vim, and then you do we do the name of the particular, let me go to this directory, the name of the particular file that I want to create. Say, for example, test data.txt. Right, it will open up this file, and on the bottom you can see this is the name of the file, and it's given the title new file. Right, now there are options here. So if I press I, the option insert comes. If I if I want to type something here, say for example, this is test data set. It got typed, right? Now, if I press escape, the insert goes away. Now, say for example, I want to replace something here. I press R, it's replaced. So if I want to replace R with say T, it got replaced with T, right? So again, I press escape and this thing goes away. Now, how do I save this data? They will save this file. Now I've created this file. I've written this particular line. Now I want to save it. The way to save it is put a colon and WQ, right? Sorry, it got saved. Now let's check this if this is present. Yeah, so it tells you that this is the data. This has been created today and at this particular time, and this is the number of bytes, of the file here. Now let's see again. Vim. So for the next times you see, you do not need to write Vim. You can just write VI. It works. VI or Vim is the same thing, right? It works both ways. If it's like this, right? Now I just open the file. I saw what is the content of the file, and I just want to close it. I do not want to save it or anything. So I do just colon Q, enter. It gets closed. Now if I say, for example, I want to edit this file. I do an insert and then I write for this course. Right. Now, if I want to do hyphen colon Q, it will not save, right? So it will tell you that no write since last changed. Either you add this this to override. So if I do exclamation Q, it will not save the changes. So, uh, yeah, it does not save the changes. Let's look at it again. It has not saved the changes. Now, if I do not use Q, I use, say for example, let me edit this. Right. I've edited this. Now I want to save it. If I do WQ, it will save. Right. Now if I check the file again, now it has saved the changes. Now, if you do not want to save the changes, the command is Q, then exclamation. If you want to save the changes, the command is colon WQ. Right. So again, basic stuff here. There's another command called nano. So nano, if I do, this is also a text editor. There are many options here, what to write, how to save, and how to exit, and so on. So this is quite meaning in case of Nano, you have all the commands written below, when you can use this particular shortcuts to do all these things. Now, say for example, uh, I want to write something. So by default, we can write this, right? For this course, advances in omics, right? I've written this. Now I want to exit, Control X. Now it will ask me if I want to save it or not. If I want to save it, I do capital Y. Excuse me. Now it will tell me which date. It's the same file I'm writing it in. So it will confirm once and I put an enter. It has saved. Now let me check it again. See, it has saved the file. Right? So text editors are basically Nano and Vim that we use on a daily basis. It's a very commonly used commands, Vim and Nano, and we used it to like either create files or edit created files. Right. So this is like we talked about first we navigating the directory, then how to create and edit files. Right. Now, what happens is in some cases you can your file is so big. So you just want to see the file, you do not want to edit or anything else. So, okay, so let's go back here. So let's, for example, uh, we want to see this particular file. Now, this is a GZ file anyway. This is a zipped file. But anyway, we want to see what is the contents of this file. This is a FASTQ file. But I won't be using VI because I do not want to edit or anything. I just want to see it. This is a big file. I do not want to edit or anything. I just want to see. So what I use is a command called less. So if I do less this, it opens up the file. Now, this is how a FASTQ file looks like. It's so you have all the sorry, all the sequences, and these are your basically your uh, quality scores for each base, right? So this is how a FASTQ file looks like. You scroll, you go below, you can see. Here you cannot use WQ or anything. You just have to use Q because you are not using, you are not editing the file in any manner, right? So you are just uh, you're just like having a look at the file, like what is inside. That's all. So like this, you can see. Now again, use Q to quit. So this is how less works. Now there are there are many other commands that are very similar to less, which is basically first is a cat. Cat is also a file viewer. So cat, since, sorry, so, sorry. So cat won't work here because this is an .gz file. Let me find this particular file, right? So let's do cat on this. So here, this is the particular file that I have, and this is the content of the file. Now, if you want to look at some other file, let me see if I do have anything else. Hmm, hmm, all are zip files. Hmm, let's try this SAM file. The SAM file should be fine. So let's do cat. See you this is the content of the file. It tells you what the content is, right? So cat is something useful, but this does not open up a screen. This like all the contents are printed here. You cannot also it's the same thing. You cannot edit here, right? So this is how it looks like. Um, mm, there are other commands. So for example, we have used CAD. We can use head, right? Head is another command that we can use. It just gives you the first few lines of the file. So the cat has given you the full file, the contents of the full file. Head will give you just the first few lines of the file. So, Right? Same thing with tail. If you want to use tail, it'll give you the last few lines of the file. So head and tail are quite intuitive. Less is basically if you want to see the whole file in a screen, and cat is basically you just want to uh, print what the contents of the file are in your screen. You cannot edit or anything like that. Okay. Uh, yeah. So there is two more, which is basically more and less. Let's try these. So this is also like less. It basically tells you first few elements of the file, and this is basically the if you see it's showing 1% of the file. Like that's all. Right. So now if you want to do it's to uh, uh, yeah, more and less we have seen. Let's try seek. So I don't think it works on this particular file, but yeah, you get the idea. But the most important one, the most kind of easy to use and most widely used is less. You do the less, you get the entire file. See, you see the difference between less. So this is the less command. One second. This is the less command. Now compare it with the cat command, right? It does not like this is not the full file, right? If you move to the right, there is the rest of the file. It just shows you the the rows according to the file, but it does not print the whole file in terms of right. Because if you see in less, this file is too big. So this is the first line, right? So it starts from here, then chromosome X, this position, this is some quality score, and this is a long, basically ID or read information, and after that the whole sequence is given. This sequence is not printed when you are using cat, right? So less is mostly used to kind of see the overview of the whole file, but you cannot edit the file. That is important, right? Okay, so these are some methods where you can use to uh, create files, edit files, and for these particular commands, cat, head, tail, less, these kind of things, you can use to view the files. You cannot edit. Right? Now, uh, like again, we come to the similar type of commands: how to make a directory, how to remove a directory, how to remove files, and how to copy files. So let's do a couple of these things. Let's go to this particular. Right, so if say for example I want to copy this particular file to this directory, so the command is cp stands for copy, the name of the file, and then the directory. So this is the directory I want to copy, and if I press enter, it will copy. It'll take a little bit time because the file is big. Yeah, it has copied. Now let's go to the directory and check. See, it has copied. So cp is the copy command, and the other command is mv, move. So for example, I want to move this particular file in the directory. So mv is move, name of the file. This is the directory I want to move it in. It got moved, right? So let's check here. See, the file has been moved here. Excuse me. Okay. So yeah, these are pretty straightforward, but these commands you need to know to kind of navigate through the whole system, because here you won't have windows where you can like click on a particular folder, it'll open the folder. It does not work like that. You have to use command line to kind of navigate yourself. So knowing this kind of commands is important, right? So yeah, and if I want to say, for example, remove a directory, let's do that. Uh, let's make a directory here, and let's move a file there. Okay, so copy a file to this particular directory. Yeah, now this particular directory has this particular file. Yeah, I want to move this particular directory. So the idea is uh, the command is rm, and if it's just a file, you just put the file name there. And if it's a directory, you have to put R, right? Hyphen R is the command, and then you put the name of the directory, it removes. See, the directory has been removed. The test directory does not exist anymore. Right? If it's just a file, you do rm space the name of the file, just it'll work. Yeah. Okay, so yeah, these are the basic commands that we use in Linux systems. Now, there are some other commands, like couple of things. So, firstly is if you want to find what are the number of word counts, right? So, yeah, if you want to find, say, for example, a couple of, what is the word count uh, of a particular file, right? So, if you go to the file, so let's try this particular file. First, let's see how many words there are anyway. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 words are here, right? Now, if you do WC, sorry, WC, yeah, see, it's given me 11. The middle one is basically the uh, uh, number of words. This is the number of lines, and this is basically the file size. Excuse me. So this tells you the number of words in a file. But if you want to count, say, for example, number of lines, let's find a file. Uh, I don't know if I have a file. Let's try this. So, snake make is basically a syntax which basically is used to make uh, pipelines, bioinformatics pipelines. So here the pipelines are basically I start with a FASTQ file. I do some pre-alignment QC, then I do alignment using this particular software which is Minimap. So these are long read data, right? So then I convert the output. The output is in SAM. I convert the output to SAM to BAM. Then I index the BAM. Then I run some post-alignment quality checks. Then I call variance, structural variance. Then I call small variance, and then I annotate the variance. So this is basically how a pipeline is written. Now I want to count the number of lines in this file, right? How do I do? I do less of this particular file, and I put a pipe, right? So I less of this particular file meaning the if you just put less and this file, it will just show you the whole file, right? But I want to pass this the output of less to WC to tell me the number of word counts, right? So for that, I use the pipe operator. So the pipe operator is basically telling you to pass the output of the command to the left of it to the right. So the less snake file, this particular the output of this will be passed on to the right command, which is WC. If I do this, it gives me this is the number of words here and this is the number of lines here. Now, if I want just the number of lines, I do WC hyphen L. It tells me these are the number of lines in this particular file. Right. So word count is using WC, and if you want to find the number of lines or number of rows, it gives you by WC hyphen L. So it's pretty straightforward like that, right? So Yeah, so unique is another thing. I do not have good files to. I I do not have any good files to kind of show the unique command, but it's same thing. So if you do, I just show you the command line for this. Uh, yeah. So if you do one thing to know, if you do and if you have any command after space, if you add hyphen hyphen help or hyphen hyphen h or anything like that, it'll show you all the commands possible for this, all the flags possible for this particular command. So uh, like this can tell you the number of lines. It can print only duplicated line. It can print all the duplicated lines and so on. So these are so many um, flags that you can use with this particular command, which uh, which basically you can use to do certain things. So the whole documentation, like what are the flags that you can use, you can get using this particular like hyphen hyphen help, right? So this you can use with any command that is there in the system, right? Okay, moving on. So these are some. So I talked about the pipe, which is like you have command one, the output of the command one, you're passing on to the command two. Right now, if you want to say, uh, if you want to save something from one file to another, say for example, let's take this file only. So I want to search for something in the file, right? So let's search for randomly Alfred QC, right? So for searching, in general in Windows, what we do, Control F, right? Here the Control F is done using grep. So grep, this is what I want to search, and this is the file I want to search in. So if I do this, it shows me the occurrences where this particular word is there, right? Now I do not want to print this in the terminal. What I want to do is I want to save it in some file, right? So what I do is I grep this, but I redirect the output to some file. So test grep.txt. If I do this, it will not display it here, but it will save it in this file. Now let's check the contents of this file. You see the contents is basically what it should have printed if you do a grep, right? So uh, this particular arrow is basically redirecting the output to a particular file or to a particular folder or whatever you want to do it. So this is also a very useful symbol that we use quite frequently while handling Linux systems. Okay, yeah, I think this is fine here. Yeah, the rest of them are like not so important. I'm touching the important parts, so that you can like if you want to start off with something with some data analysis using genomic data or something else related to omics data using Linux commands, you can get started because these are not so difficult, right? So these are like commands that are if you start working on these on a Linux system on a regular basis, you'll get used to this command. So it's not that difficult. So yeah, grep is um, very widely used. Grep is basically the control F for Linux, right? For now, there are other. This is one of the important ones, which is sed, which is the stream editor, which kind of can replace or um, remove certain things from your file. So sed help, right? So here, these are all your commands that you can use with sed, right? So if you do, let's see. Sorry. Mm, mm, okay, let's see here. Give me just a second. Sorry for that. Yeah. So set can be used to, say, for example, replace something in your file. So, for example, let's go to that file, this thing. Now, say, for example, we want to uh, replace, say, for example, sample with sample one or something like that. It's just an example, right? So that command would look like sed. Then you have this inverted comma. S is basically telling you to replace, say, for example, old. This is the old thing that we want to replace, and this is the new thing we want to replace it with. Right? Sample with sample one, and then we use the and we close the inverted comma, and then we put the file name. Right? See, they have replaced sample with sample one. Right? Sample with sample one. Right? If it is connected with something, then it will not replace, right? So if it is only a simple one and then it is not connected to anything else in the whole line, so if it's only sample, it'll change it. So this is how it is done. Now, if you want to again, if you want to save this file, you do a redirect to another file. So let's name this modifier, and then it is saved here. See, it is saved here. So right. So sed is also something that we very widely use. So sed and grep, I think, will be uh, very important. Another thing is that the star operator is very interesting. So let's, for example, go to this directory. So here are two files. First view, we have two files here. Now let's assume that my list of files in the directory is very large, and I cannot find this particular files. And I just want to list the FASTQ files in this directory. So what I do is I use a star ls. Ls is always used for listing. Then I use a star, and then I use, say, for example, FASTQ and then star. So the idea is FASTQ should be present in the name of the file somewhere, and I want to list those files. So if I do that, see, two files are getting listed. So star is something very widely used where you kind of fill in for, like, you do not know the whole file name, or you do not want to give the whole file name, but you use star to kind of give a pattern. So this is the pattern that FASTQ should be present in the name, and then use stars on both sides to tell you it can be, there can be other things on the left and the right. If I just use left wala fast star and not right wala star, it will not give me anything, because it means that it ends with FASTQ. But if I do with this, it'll give me these two, right? So a little bit, you have to be a bit careful of how the files are named, but this is the general idea. You put in star where you do not know what the rest of the thing is, but you have to put the stars carefully, because if you put it like this, it will assume that the end of the file is FASTQ. But in this case, the end of the file is fastq.gz. So this is also quite widely used. Okay, so yeah, now another couple of things that are important is basically uh, I will come to SCB first. Let's get to wget. So there are other commands for wget as well. So basically, it is used to download something from FTP sites or directly uh, from the internet. So let's try to download some VCF files. So I go to this particular, sorry, let's go to the FTP directly. Yeah. So this is a site where you have all the basically chromosome-wise VCF files. So these are VCF.gz files, chromosome-wise variant called files, and I want to download a few of them. Say, for example, I want to download the chromosome 2. I want to download this file. So how do I download? I can directly download in my system. If I click here, it will be downloaded in my current system, but I want to download it in my Linux system, right? So that this is not the current system that I'm working in. So I copy this link. I go to my Linux terminal and I do a wget space the link. If I do this, it gets connected and it gets started to download. Right. So will not take much time. The speed is high, but this is the name of the file, and this is getting downloaded like this. So wget and curl, both of them, right? Curl is also very widely used. So if you want to download something from the internet, curl is also very widely used. So you can use curl as well. But wget and curl, the idea is same, that you want to download something from the internet and basically a file, basically from FTP sites mostly, but you can use other. Say for example, I want to download some GitHub repository. Say for example, this is a repository. So this is a repository that we use commonly. So this is used for structural variation annotation for humans. Now I want to download this particular repository. So if I do this, if I just copy the Git address and I use, I'll cancel this because I do not need this file. And if I use wget in this, it gets connected and it gets downloaded. Right? See, the Git file has been downloaded. Or you can use for Git, mostly use Git clone. So if you do Git clone and then name of the repository, it'll clone into the folder itself. So yeah, see, this particular folder has been created inside this. This is the whole GitHub thing. GitHub data has been transferred here. So if you want to download any normal files, wget curl is a file. If you want to download GitHub softwares, git clone is the way to go. But in my case, my system has these things installed. Yours might not, so you need to install these first, then you can use this. Right? So be careful about those things as well. Okay. Yeah. So these are all the basic Linux commands that I wanted to cover. Other than that, I wanted to cover a few other things, some softwares that we use for variant calling and alignment and so on. So Sir talked a little bit about BWA. So I just want to kind of show you how the BWA commands work. So uh, let me go to my system. So this is a HPC cluster from my department that I use. So all my softwares are downloaded here. So I just load this module. So uh, this is the software, right? This is alignment via Burrows-Wheeler transformation. This is the name of the software. So if you want to take a look into it, this is the GitHub repository, but I guess you'll not require the GitHub repository. Rather, you can. Uh, this is also not very good. Let's see if we have the paper here. Mm. So again, this is for short read. If you see in the GitHub also, Minimap 2 is being used for PathPio and Nanopore because these are long reads and it retains all BWME features and so on. So BWME is basically for short read, mostly Illumina sequencing data and so on. Let me see if I think this is the paper. Yeah. So this is the Burrows-Wheeler alignment algorithm paper. This is the algorithm. I don't know if you remember or not. We talked about suffix trees and so on. So this Burrows-Wheeler transform is a very interesting thing. If you want to give it a read, please do. Yeah. Yeah. So this is how the command looks like. So if I just do BWA it brings out these are the commands that I can use. So for the MEM algorithm, which is the alignment algorithm, so we use BWMEM, right? So we do BWA, MEM, and then enter. So these are all the uh, commands that you can use for BWA, MEM. So you can specify number of threads, basically multi-thread processing. If you want to parallelize across multiple threads, you can do that. But the main thing is the command is this. You do BWA MEM, then you give these options if you want to say, for example, increase the number of threads. You want to give some scoring information as well. You can do some smart pairing and so on and so forth. This keeping of the alignments and so on, so on. So these are like extra flags that you can give, but the main one is BWMEM. Then all the options that you want to give. After that, so this is basically the reference file. So the idea is that the alignment will take place that where there is a reference and there will be a FASTQ files provided. So these are two FASTQs. So in Illumina we have read one and read two. So read one FASTQ, read two FASTQ, and this will basically be your reference file. So yeah, so this is how it looks like. And another software I want to, meaning like this is how the commands will look like. So when you are doing the alignment, you come to the terminal, you do BWA MEM, and then you provide whatever options you want to provide, and then you provide the reference file and the FASTQ file, and it will do the alignment for you. Right? Another thing I wanted to show you is the GATK module. Uh, uh, so the GATK is basically, let me show you here. 1 second, it's loading. GATK stands for basically Genome Analysis Toolkit. So this is a set of softwares in one wrapper where you can use multiple things together. You can use alignment, you can do marking duplicates, you can do variant calling, you can do structural variant calling. So this is all compressed into one particular module. I don't think this is opening. Excuse me. Oh, let me show you in the terminal only. So if I do GATK, I think 1 second. The other GATK version will be better, I think. Yeah. Uh-oh. This is not well installed. 1 second. Yeah. So these are all the things that you can do. So it's a very long list of things. Like, yeah, we have to scroll 3 pages. So starting from base calling, mostly using short read. GATK is for short read, not for long read, or not for ONT or tag biodata. So yeah, you can use for trimming bases and try copy number variant discovery. You can do coverage analysis, depth of coverage, breadth of coverage, and so on. Quality control, different duplicates you want to mark, you want to check read group sums, and so on. There's a lot of things you can do here. Uh, uh, Flow based tools, so basically flow cell based stuff you can use. flow feature mapper, and so on. Genotyping array manipulations, can use for metagenomics data as well, deep data manipulation, and so on. This is a very long process. You can manipulate the reference bases on. You can do short variant discovery. Haplotype caller is the most widely used short variant caller for germline and SNPs and indels, right? And then you can do structural variant discovery and so on and so forth. You can do a lot of stuff using GATK, right? So these are the kind of tools that we use for processing the data. We use this in Linux commands only, and basically just wanted to give you an overview about the kind of in real world, if you want to do analysis with any sort of data, how would you go on about it. So first you need to install this. So if you want to install, you go to the GitHub page. Oh, this has opened GATK. So if you are interested, you can take a look at here. Uh, yeah, so as I was saying, uh, yeah, if you want to run this in command line, you can use the commands as I showed you. For example, for BWMEM, similarly for GATK, you can use these command lines in Linux environments and run your samples for data analysis, right? So I personally work on human data, and I am not very familiar with other kind of there are basically other kind of tools for handling, say for example, yeast data, microorganism data, or some rat, mouse data, or something like that. But for human data, these kind of tools are widely used. So, yeah, wanted to give you an overview of how to run it. So these will be big data sets that you have to run. So this will take time. So it won't. I cannot do it in like 5 minutes. It will take time, a lot of time, an hour. So, for example, BWMEM, if you give it the whole genome sequencing FASTQ file, it'll take 30 to 40 hours to align the whole for one sample, right? For align all the reads for one sample. So it's a long period of time. So yeah, I just wanted to give you an overview about how these things look like in Linux environments and so on. And as I showed you the snake file, if you remember, the kind of flow looks like that. And if you want to build a pipeline, a bioinformatic pipeline, starting from raw FASTQ files, raw read files, and kind of going down to variant calling and annotation and so on, you can build pipelines using these kind of softwares. And as I was saying, there are other types of organisms as well, and there are other tools that are specific to those organisms that you can use. I primarily use human data, so I'm more aware of these things rather than those things. So if you want, in the next class, I can maybe give you a couple of examples of those as well. Okay, so yeah, this is done here. One thing I want to touch before going on to the questions is basically uh, okay. So this, I think Sir discussed about BLAST. Yeah. So BLAST is basically a sequence alignment software. So you can run it on the web tool as well. So if you go for nucleotide BLAST, you enter a sequence or you upload a file, and then you choose the databases. So there are a lot of databases for nucleotides. You can choose it from here. You can add the organisms. Say for example, rat. If you type something or human, yeah. So you can get the organisms here, and then you optimize for highly similar sequences or most dissimilar sequences or those kind of things. Then you click on BLAST. It will give you an alignment of your sequence with multiple other reference genomes, and you can do it in on the internet only. You do not need to you do not need to kind of download this to to run it on your system. So Blast is something that is again very widely used, but a little bit old. So we have much better softwares now, so we do not use Blast. But yeah, this is the one that I was looking at. So yeah, this uh, particular blog from UC Berkeley, I think, gives a very good comparison of different types of blasts. So one of them is blast 10, basically comparing two nucleotide sequences. You can compare a protein sequence to a nucleotide sequence as well. So this is a translated nucleotide sequence searched against a protein sequence, which is BLASTX. And T-BLAST10 basically protein sequence searched against a translated. It's the opposite of BLASTX. And then you have protein BLAST as well. So amino acid sequences compared against each other as well. So there can be a lot of, you can try try doing this. This is a good exercise to do if you want to learn about sequence alignment and so on. Sorry. Sorry for that. Yeah. So yeah, you can take a look at that. The other thing that was covered, I'll quickly cover that, is the SRA database. Right. So this is sequence read archive where you put all the data here. Say for example, mm, let's search for something. Let's say 1 kgp AG001. Let me click it or not. Yeah. Mm, no, let's try 1 kgp. Yeah, so this particular sample, so for example, this is the 1 kgp is the data source, which is the 1000 genome project. From there, the data has been acquired here. So if you click on that, you get all the information. So biosample ID and SRA ID, these are your identifiers. Organism and like classification of organisms. What is the cell line, collection date, sample ID, and so on. So this is the bio project. So if you go to the bio project, it'll give you the project information. So this is the project information that you have to, in this particular project. They study duplicated genes. Yeah, you collect used a complete T2T genome sequence to identify 213 specific gene families. From these 32 paralogs were found in most human and rice and mice and so on. So this gives you the project description, and this has originated from UC DVs. The biosample, SRA data, sequence data, what is the uh, size of the data? So 957 gigabases of data. And basically, the bioproject information will contain all the project information, right? So, and when and what, how many SRA experiments have been done, and around 450 samples are there, right? So, yeah. So this is the accession and this is the ID for bioproject. So, so for example, you want to access some of the data. Uh, so you need to do not do it from here. You do it from some other sources, some process data. Say for example, you go to gene expression omnibus. So this gives you mostly in gene expression omnibus you have RNA sequencing data, right? So if you want to, and there are GSC IDs for these data sets. So let's try this particular. Randomly, I'm putting 4 ones. I don't know how this is there. So this is a GSC ID. So it tells the title will tell you what it is doing, experiment type, which is this microarray data. This is not RNA sequencing, but this is gene expression data, but microarray. So this tells you the summary of the whole project, overall design of the project. Then so Geo2R is basically an inbuilt software for Geo, which you can use to analyze the data here only. You do not need to download. Or you can, yeah, you can check the samples. What are the sample details? You click on the sample name and it will give you all the sample details, right? What type of labeling and those kind of things, protocols, when it was collected, and values and so on. You can collect. you can download from here, or you can download series matrix files. Yeah, you can download, say, for example, this particular file. This is the actual data file. Right? So if you open the series matrix file, not Word, not Word. Yeah, this gives you all the information. These are your sample IDs, and yeah, so these are all your gene expression data. The ID ref is basically the gene reference. So these actually map to some genes. One is some gene, two is some other gene, so like that. So this is RNA, not RNA sequencing. This is a microarray, but gene expression data. Right? So you can get from here as well. Sorry. Yeah. So that is all that I wanted to show for today. There are some MCQs from last year's assignment. We'll discuss that. And yeah, if you have any questions regarding these, you can, like, if you want to practice some of these, please use the link that I gave below for the Google Drive. I think, yeah, this is the drive. This has mostly all kinds of files that you can think of. Excuse me. You can work on these files if you want. So another thing here is the FastQC, which is another program which uh, tells you the quality status of your FastQ file. So I think I might have some FastQC files here. Uh, yeah, so this is how a FastQC file looks like. You have data quality, per base sequence quality, per sequence quality scores, GC content, and so on. So I think we discussed this a little bit, but this is basically quality control of your raw read file, FASTQ file. So this is another software that is very widely used, and this is very old, but still very much in use. So yeah, I think you can look at these resources, and yeah, if you have any problem, you can let me know. I'll try to help you. Other than that, then we can move into the questions. We do not have many in terms of, like, yeah, this will be more straightforward. Let's move on. Okay. So Bowtie 2 Barzola transform that I showed you. Bowtie 2 is another software that we use for aligning, and Gem 3 also. So let me show you Bowtie 2. So this is the Bowtie 2 GitHub. Uh, it's similar in terms of how you can run it. So give Bowtie. So this this is the FASTA file. These are your two FASTQ files. So that is how you build. That is how you run any kind of aligner software. So BWMEM I showed earlier. This is Bowtie 2, and the other one is L3. If I you know yeah. So this is also used for doing sequence alignment. Here also same thing. You have the reference genome. You have to index the reference genome, and yeah. So this is your command. This is your basically reference file, and these are your fast. This is your FASTQ file, and this is your output file. Excuse me. Sequence alignment map. So another version of the aligned output file is BAM file, which is a binary format of the same SAM file. You cannot. It's not human readable, but the size is significantly lower. So we store mostly the data in BAM format. But SAM is the main output. You need to convert it to BAM. Right. So okay, moving on. So firstly, why we do this trimming? Because the the exact read might not align with the reference genome. So for that, probably you need to trim the ends of the reads. Primarily, it might be adapters while you're sequencing, and you have gotten that adapters in your read data as well. So you do not want that. You've got to trim that, or it's extra pieces of DNA which are not getting aligned to the to the reference genome. So you need to trim that. So for that, it is cut adapt is the tool that is used. Yeah, it, it removes adapter sequences, poly A tails, and other types of sequences, unwanted sequences from your high throughput sequencing leads. Uh, uh, one second. Yeah, yeah. So you give cut adapter hyphen A. What is the kind of reads that say for example you want to read the adapter sequences, so you give the adapter sequence here, what is the output and what is the input. So this is how it runs it. I hope that's clear. Okay, so sorry. So as we discussed earlier, the answer would be like, so you're trying to query a protein sequence against a translated nucleotide database, that is T-Blastex. Again, going back to this particular log that I shared. Yeah. So translated protein searched against translated nucleotide searched against a protein sequence, and the opposite of that, protein sequence searched against a translated nucleotide. So translated nucleotide versus protein. So that is what you want here, which is T-Blastex. Okay. Sorry. Okay, so for variant annotation, we use all three: Alamut batch, VP, and ANOVA. VP and ANOVA are more widely used. These are VP is ensemble from ensemble. Uh, so this is the variant effect predictor. Install the databases for this, and provide your variant called file. So VCF file you have to provide, and then it'll annotate the variants in the VCF file. So VP is one of them. ANOVAR is another. This also is quite widely used. So this is the again gene and region based annotation. It can do all. And what is the one? So, sorry, but this is not free, right? So this I think you need to uh, do some. I think you need to buy or take subscription for this. Then you can run. Uh, this is not exactly free, but the other two, ANOVAR and VP, are free. So those two are more widely used. In this case, the intuitive answer would be it would be printed 100 times, but there is a syntax error here. You have to put a semicolon before do. Sorry, before do you have to put a semicolon. So there is a syntax error here. So other than if you put a semicolon there, it will print 100 times. But since there's a syntax error, this will give you an error. This will not print anything. So none of these is the answer. Okay, moving on. Okay. Okay. As we discussed, the answer is the pipe operator. So for the pipe operator, you have on the left command, whatever the output of that is, you are passing out to the right command as input. So for that, you use the pipe operator. So if you want to run programs sequentially, the pipe operator is used. Okay. Uh, The answer to this will be Vim, Emacs, and Nano. Vim and Nano we already discussed. Emacs is also there. It's similar to Vim and Nano. VS Code is a software from Microsoft that we use for coding. So that is certainly not a command line text editor. So other than that, the rest of the options are correct. The answer to this is basically this the last option basically. So you do find type D is basically all the directories in your file, and then you copy this to file.txt, and then you sorry, copy the file.txt to the rest of your to all the directories in the current directory. So you have to give the file name here, before that cp, and before that you have to find type D exec. So that, and then you do file.txt. That curly bracket tells you that all the directories, and after that you put a slash. So this is fairly simple. We discussed curl and duplicate. We already discussed, but the rest of them are also there. I do not have them installed here, so I'll just show you on the internet. Yeah. Uh, yeah. I think Yeah, I think this is not, this is paid in some way. I'm not sure. I've not used this. But curl and duplicate are basically the main ones to use. Prefetch is also there. Yeah, so this is how it is used. Link, del as prefetch, and then you do the HTML logging. So yeah, this is the content, but generally we use curl and wget. Those are the most widely used and very simple. You do wget the link, curl the link, it will download. So yeah. Okay, so something about the FASTA file, it always starts with that greater than sign, right? So if it is at the start of the line, you can use both. So you can use, so what this means, the upper arrow means it's the start of the line, and FASTA file starts with that arrow. But even if you do just this, it'll also give you the output like this, because in general the FASTA file also starts like this. So here you're specifying the start is like this. In this case, you're just telling where this is. So generally, it is in the first. This is the first element of the character of the FASTA file. So both of them works. And grep is, as I told you, it's basically a control F uh, for Linux commands. So yeah, both of these work. This is also very straightforward. One I showed you uh, in the class. So either of hyphen hyphen help or hyphen h works. So you do this again. Let me go back here. You do bwa nem hyphen hyphen help. Sorry, I think yeah. It works like this. So either of them works. So yeah. Okay. BAM stands for binary alignment map, not binary alignment file, and it is basically a binary file for the SAM file. SAM is the sequence alignment map. There you can it's human readable. You can read the reads and how it is mapped. The binary file for that is BAM. So it is obtained when raw reads are mapped into the genome. Sorry, it is obtained when the raw reads are mapped to the genome, and it can also be obtained from a SAM file. So basically, you convert the SAM file to a BAM file, which is the binary version of the SAM file. So it's the information content is same for SAM and BAM. Just the one is human readable, one another is not human readable. It's a binary file and it takes up less space, so we use BAM file. So generally, the sequencing data is stored in the FASTQ format, but 454 uses this SFF format. Right? Mm, yeah, I think yeah. So this is the format that is used for by Roche 454. So, but in general, Illumina and the rest of the sequencers, they store data in FASTQ. So for Roche, it is SFF. Okay, as I shown earlier, it actually contains all of this. Let me quickly go back there. Yeah, this is the bioproject page. It contains everything, the description, the grant, the submission, registration date, everything. So publication, project description, grants, and everything. It contains everything. So all of this is the correct answer, but also you can put all of them as correct as well. Okay. Yeah, this I have not put, but anyway, let's discuss this. Under biosample, also everything is mostly these things are available. So biosample contains all the important things about the sample information. So what tissue it is in, it has been collected, what organism, which types of cell types have been collected, and so on. Sample name, and also common name of the organism. So under bioproject, the project details is contained. So your grants, your publications, and those things. And biosample, all the sample details. So it's quite straightforward. Yeah. So if you basically convert one type of genomic coordinates to another, say for example, moving from GSH 38 to 37, or moving from GSH 38 to, say for example, T to T, the tool to use is liftover. So UCSA liftover is basically this. It lifts over the coordinate of one build to the other build. So liftover is the answer. So in the UCSC browser, you'll have expression track, EQTL if the variants are expression quantitative trait loci. It'll have the SNP track. It will not have the homolog track. Basically, the homolog track will tell you other uh, s- species information. So that won't be there in UCSC. So the answer is homolog track for this particular question. You can like uh, kind of explore UCSC a little bit because that is kind of interesting. So yeah. So again, these are different types of bed formats. Sorry, file formats. Bed is the easier format. So you have chromosome, start, and end. So that is the entire description of the bed format. Three columns in a text file. That's all. A BAM is all something that we've discussed earlier, and Big Lolly is another format from uh, UCSC. It's a big bed file. This is another type of format from GCC that gives an output in. So probably take a look here as well. Okay, this I have not put here because I was also not too sure. MOBA term is not the correct one. The one that I gave you is MOBA X term. So uh, yeah, this is the MOBA X term. This MOBA term is not there. But the other two softwares are Putty and Kitty. So yeah, these are the software names you might want to remember. Hmm, yeah, that's the final one. So two different tools can be used for variant calling. One is a fee base, and another is a mutex tool. So these are also quite widely used. Let's show you. This is fee base. Uh, this is quite new and kind of describes this. Uh, look here, and the other one is, so let's do it here only. Mutex. It is a GATK module. So take a look at this. Okay, so that's all for today. If you have any questions regarding this, you can ask, or we can close the session for today. Excuse me. Okay, if we do not have any questions, we'll close the session for today. See you next week, Tuesday, same day. Thank you for joining. I hope you're here in the call, or like, I don't know, you have not replied to anything. So yeah, thank you for joining. See you next week. Yeah, thanks.